Hey everyone, I am here to welcome you to our first remote week of learning. I'm going to walk you through a little bit what I'd like you to do for this week's lecture, which includes physical and chemical methods of microbial growth. All right, so before we get to the lecture, I just want to bring up a couple of things. The first is your course Blackboard page. And so on the Blackboard page, if you go to our class, um, you can find the revised syllabus on the course homepage, which is right here. So the syllabus will update you on new lecture policies and attendance policies and grading policies more specifically since everything's changing so much. If you go to the lecture materials tab, what you'll find in Unit 3 is you will find over here, you'll find a pre-class reading assignment. Instead of giving you a paper assignment, I'm doing it through McGraw-Hill. It's called an adaptive learning, an adaptive reading assignment through LearnSmart. So this is something that should take you about 20 minutes, and you should do this before you do the lecture. And then when you go to the lecture, you can click on physical and chemical methods. You can open out either the PowerPoint or the PDF. And this will give you the full lecture. The PowerPoint, of course, will have my notes in the notes section, which can be helpful. There is a link to a summary chart, and you may have to click on this right here to open up the summary chart and to download it. If you can't print it off, maybe you could just sketch a copy of what the summary chart looks like. So for today's lecture, those are some of the things you're going to need. I'm going to go ahead and show you what some of those things are. So don't forget that you'll also want our course pack. And so our course pack opens with the Unit 3 lectures. Physical and chemical methods of controlling microbial growth will be part of Lecture 1. I've made it a single PowerPoint, and next week I'll post a PowerPoint for the antibiotics, which is another part of this same lecture. And so for this lecture, we're going to be identifying ways that we can control microbial growth. And I'm going to give you some scenarios, and you will pick methods that you could use in those different scenarios. So these first two bullet points right here are what we're really going to focus on this week. And then as you go through the lecture, while I'm talking or just individually on your own, going through slide by slide without my voice, make sure you fill out the notes as you go along. So these notes right here, just fill in definitions as you go through the slides. So today we're going to cover pages 57 through 58, 59, 60, and 61. And when we get to antimicrobial methods, that's where we're going to stop today. So you should be filling out pages 57 through 61 in your course pack. Okay. Another thing you're going to want handy. So you're going to have your course pack notes. You'll also want that supplemental chart, the one that I just showed you on Blackboard. And this is a summary table to compare different methods of physical and chemical methods of controlling microbial growth. And so this summary chart, you can fit almost everything you need to know for the test on this one chart. And I will give you examples of how to fill out this chart and what all these different letters mean. Okay, so we've gone over our new syllabus. We've got your lecture course pack ready, and hopefully you also have that summary chart ready. If you have all of those things, let's begin. And we're starting today with some discussion questions. Some of you all may have noticed that in your lecture course pack there are 3 bullet point questions, and I also created, in place of an in-class discussion, I created a discussion board. So if you go to Blackboard, instead of going to lecture materials, you'll now want to go to the discussion board. And here on the discussion board, there's already 16 posts from 9 different people, so some of you have already done that. But if you haven't done it so far, what I want you to do is stop the recording right now, go to this discussion board, and post at least one thing. There are 3 different threads that I contributed. One thread says, can you describe a scenario where you might want to kill microorganisms? Or describe a scenario where you want to promote microbial growth. And then finally is, can you think of a scenario where you might just want to prevent microbial growth or remove microbes? So post your comments on one of these 3 threads, or you can create a thread of your own. And then let's continue on. Read what some of the other students wrote, comment on their comments, or just write your own independently. Okay, and so we want to control microbial growth in a number of different ways. We can kill them, we can prevent their growth, or we can remove them. And then in other instances, we might want to promote growth. And so I'll look forward to your comments on that discussion board. For this week, we're going to be focusing mostly on physical and chemical methods of microbial control, a little bit mechanical methods. And so just to distinguish between these 3 things, if something is a chemical method, then that means you'll find what it's made of on the periodic table. So something like chlorine or bromine, all of the different complex chemicals that are in your Lysol wipes, those are made up of things that are on the periodic table. So we would call them chemical agents. A physical agent will be something that will involve either heat or radiation. And so these aren't, substances that are chemicals on the periodic table. And similarly, we have mechanical methods, which are also methods that are used for decontamination. And so a mechanical method will be something physical, not like heat or radiation, but something that involves a scrubbing or a filtration or a mechanical removal process. And so physical and chemical agents can be used to disinfect or sterilize. They can be used as an antiseptic in some cases, or they might just be used for sanitization. And we'll go over the difference between all of those terms. A mechanical method, these will be used for decontamination. So we're going to keep track of all of the different methods in the upcoming slides and what we use them for. Okay, so let's start with some of that basic terminology. When we see the term germicidal, this suffix right here, cidal, that means to kill, right? So if you are homicidal, you will kill people. But germicidal means we're going to kill microbes or germs. You know, I don't like using that word germs, but it's a very general term for all microbes. All right, so in bactericidal methods specifically, since cidal means to kill, bactericidal means to kill bacteria. On the other hand, something that is static means, stasis means doesn't change, right? So something that is bacteriostatic as a whole will simply just inhibit bacterial growth. So a bacteriostatic method inhibits bacterial growth. All right, so something that's really harsh is more likely to be bactericidal, but something that's more mild might be bacteriostatic. All right, fungicidal means to kill fungi, whereas fungostatic means to prevent fungal growth. So another word for inhibit would be to prevent. Okay, and then finally, sporicidal agents are used to kill bacterial endospores. And so if we review what an endospore is, it's this dormant cell structure found inside certain types of microbes. So for example, this microbe could belong to the genus Bacillus or the genus Clostridium. These are the 2 bacterial genera that form endospores. And what we remember about endospores is that they form under harsh conditions, they're really, really resistant, and they are therefore very tough to kill. And so, a sporicidal agent will be something that can be used to sterilize. That's supposed to say, whoa, kill right there. A virucidal agent is something that can be used to inactivate viruses. And here we have our coronavirus right there. Let me fix that for a moment. Okay. Let's see if we've got this fixed. Okay. All right. So I think that takes care of a lot of our terms on our study guide, right? So far we have covered bactericidal versus bacteriostatic. Sporicidal means kills endospores. Something that is sporicidal, I'm going to note this here, is something that will be able to be used to sterilize. If something can kill endospores, then it can kill everything. All right, and we don't want to say that virucidal agents kill viruses, since viruses technically aren't alive. Rather, virucidal agents are those that inactivate viruses. All right, so moving on with our terminology, we mentioned that sporicidal agents can be used to sterilize. So one important thing is to know the difference between disinfection and sterilization. Disinfection refers to the destruction of most microbes, whereas sterilization refers to the destruction or complete removal of all microbes. And so that means bacteria, viruses, fungi, and bacterial endospores, which are those toughest things to kill. All right, another thing that these two things have in common, though, is that they're used on inanimate surfaces. And so you can't disinfect human skin because human skin is an animate surface. And so we're going to distinguish between disinfection and antisepsis. Disinfection and sterilization are terms that we use to refer to only objects that aren't living. So your kitchen counter, a doorknob, a loop that you're going to flame sterilize in the lab, that is something that can be sterilized. All right, so disinfection and sterilization, what they have in common is that they kill microbes on inanimate surfaces. But disinfection kills most, sterilization kills all. So if you have a product that gets rid of 99.99% of microbes, then that would be a disinfectant. So a disinfectant refers to the product, whereas disinfected refers to the adjective of the object that just you did the disinfection on. Some things that can be used to kill endospores and all microbes, those agents would be referred to as sterilants, and then the surface that you just sterilized or used a sterilant on would be called sterilized. So sterilization is the process, sterilant is the noun, sterilized is the adjective. All right, and so when we're talking about something that we use on living tissues, this can be skin. It also could be the inside of your mouth, like mouthwashes. So if something is bactericidal, so antiseptics, because they destroy or remove most microbes, these are going to be things that are bactericidal. So they usually have something in it that is an active ingredient, something that's antimicrobial, right? So regular hand soap isn't an antiseptic. Something like salt water wouldn't be an antiseptic because those things are not bactericidal. So antiseptics have chemicals in them that are bactericidal and they're used on living tissues. So it's similar to a disinfectant, but it is different because we're using it on living tissues. Some things like alcohol, right? The alcohol found in hand sanitizer. This is something that can be a disinfectant if you're using it on your kitchen counter, but if you're using it on your hands, then we would call it an antiseptic. So the process is antisepsis, The product that does the antisepsis is called an antiseptic. In sanitization, this is a general term. It can be used for either living tissues or objects. So it can be used on animate or inanimate surfaces, and it refers to the process of reducing the number of microbes to safe levels or sanitary levels. So there's a little bit of overlap between sanitize and antiseptics. For example, hand sanitizer, you might think that's a sanitizer. But technically, since it has that bactericidal agent in it, it's really an antiseptic. So sanitization or products that are sanitizers are often going to be things like regular soaps with no antimicrobial in it. All right, so regular washing of your hands with regular soap, regular washing of dishes with regular dish soap, soaps that don't have these antibacterial ingredients, this would be sanitization. Okay, so now we're going to move on to different modes of action. And so going back to our course pack here, I just want to bring this back up. We have covered sterilization versus disinfection. We've covered antisepsis and sanitization. And then decontamination just refers to any of those mechanical methods that involve scrubbing or filtration. So we'll see some of those as we go. When we get to modes of action, we're really just referring to how one of these agents works. And so when I refer to an agent, I mean something like bleach or vinegar or ammonia or heat or radiation. So the agent is the thing that's doing the killing or the preventing of growth. The mode of action means what does it do so that it's actually working? And so most of the physical and chemical agents that we're going to talk about are going to destroy bacterial or microbial structures. So if an agent like bleach damages all cellular proteins, this is very harsh. To denature a protein specifically means that it's unfolding right here. So to denature means to unfold. And if a protein is unfolded, it doesn't work. And if all of the cell's proteins get denatured, the cell will die. And therefore, agents that denature proteins are bactericidal. There are certain agents that will break up cell membranes. So we've got these surfactant molecules right here that are amphipathic. And amphipathic molecules are partially hydrophilic and partially hydrophobic, just like membrane lipids. So they kind of lodge themselves inside the membrane lipids and then eventually break them up. So certain modes of action or certain types of chemicals will be that they damage or break up the cell membrane. A third type of mode of action will be if the DNA is damaged. So certain things like radiation will damage DNA. And so that's what we see right here. If DNA is damaged, the cell will die. So these three modes of action right here are modes of action that will lead to an agent being bactericidal. A fourth mode of action that's actually bacteriostatic will be if the cell gets dried out. So dehydration is when the cell loses its water because of osmosis. So if it's in a really salty or sugary solution, the water will get sucked out of the cell and the cell won't grow anymore. And so methods that involve dehydration are going to be bacteriostatic. So as we go through our physical and chemical methods of microbial control, you're going to be expected to know what is the mode of action of this method. Or in other words, how does it work? And I'm going to limit it to you knowing whether it's one of these four things. And so these are the only four modes of action that you will be responsible for knowing for our physical and chemical methods. These ones right here. All right, so before we get into the specific methods, I want to talk about things that we think about when we're choosing a germicidal agent and things that will affect how effective that agent is. So the first thing is the number of microbes present. So you can probably imagine that if there's more microbes present on a surface, then the agent is going to be less effective. All right. If there are biofilms present, or if the microbes form endospores, these sorts of things are going to be harder to kill, and so the agents will be perhaps less effective. The concentration of the agent. This one is an important one. Some products have an optimal concentration, so it's not going to be more is better. So, for example, alcohol. In alcohol, 99% alcohol is not effective because it evaporates really quickly and it doesn't get inside cells. So instead of using 100% or 99% alcohol, we try to use alcohol between 60 and 70%. And this is because it'll stay on surfaces longer. There's more water mixed into it, so it won't evaporate as much. And also, that little bit of water that's mixed into 60 to 70% alcohol is going to help that alcohol get inside cells through the aquaporins. And so whenever you're using a chemical agent, make sure you follow the package's instructions and you want to mix it to the optimal concentration. So always just follow the directions of what's on the package. The mode of action will also affect how effective something is. So for example, something that denatures all cellular proteins is going to be more effective against germs than something that dehydrates them or something that mildly damages their DNA, like UV radiation. So some agents will have much harsher modes of action, and the ones that break up cell membranes and the ones that damage proteins are probably going to be the harshest and the most effective against bacteria. They're also going to be the most toxic to humans because of that harshness. Exposure time to agent. This one is a simple one. The more time a method is used, the more effective it is. And so if you are washing your hands with soap and water, especially if you are trying to scrub those hands and get rid of any coronavirus that might have landed on them, the CDC recommends you use hot soap and water for 20 to 30 seconds. So you really want to make sure you follow the instructions when you're using something. And if you're using a disinfectant on a surface, the package will often say to let it sit there for 3 to 5 minutes, for example. The Rocal you put on your bench, you're supposed to spread it around and then just let it sit there for a while. And there are other things that affect agents as well. So for example, temperature. Hot things will evaporate. And so if you are using alcohol on your kitchen surface, or if you're using it at hot temperatures, it's going to evaporate. So temperature might affect the effectiveness of certain things. The presence of organics can also inhibit how effective something might be. So if a scene is contaminated with a lot, when I say organic matter, if there's a lot of blood and fecal material mixed in, then that means your agents aren't going to be as effective and you'll either need more or you're going to need to expose those surfaces for a longer period of time because the agents are going to be less effective in the presence of that organic matter. All right, so we're ready to move on to our individual methods of physical and mechanical methods first. So we'll start with these. I'll go through a couple of them. I'll show you how to fill out your charts, and then we'll move on to the chemical methods after that. And so if you look at your study guide, I'm going to jump all the way from the physical and chemical methods, and I want to move to where your study guide is or your lecture review guide. So after your terms, on page 65, we have your concepts. And one of the things I want you to note is that for all of the physical and chemical methods, you're going to need to know the mode of action. You're going to need to know if it's bactericidal or bacteriostatic, if it's used for disinfection, sterilization, sanitization, or antisepsis. Or some things are going to be used to preserve. So to help you with this concept, I've prepared a summary chart that walks you through these things. So if something is bactericidal, we are going to give it a code. I believe it's going to be BC for bactericidal. Or if it's bacteriostatic, we're going to give it the code BS. If it's a decontamination method, that means it's a mechanical method, and we'll call it DC for decontamination. If a method can be used to sterilize, we're going to say ST for sterilize. And of course, something that's a sterilant can be used to disinfect, like bleach. If it's more dilute, it'll disinfect rather than sterilize. But if something can be used to sterilize, then we'll say yes, ST for sterilization. If it can't be used as a sterilant, then we'll give it a D for disinfectant. If it just sanitizes, then we'll call it SA. Or if it's preserves, then we'll call it P. So something that's bacteriostatic might be used to preserve foods. And then also we'll take a note of what the mode of action is. And again, it's going to be one of 4 different things. Okay, so go through the following slides. Take notes in your course pack and as we're going along. So the first one that we've got is heat. And so there are various types of heat methods, but no matter what all heating methods have in common is that they're definitely bactericidal. The mode of action for all heating methods will be that they denature or damage all cellular proteins. All right. And whether it's used to disinfect or sterilize is going to depend on the method. And so dry heat could be something like flame, or it could be something like incineration. All right, so when you flame sterilize your loop right here, you're sterilizing. So dry heat can be used to sterilize. Moist heat just means a heating method that will also involve water or H2O. So there are different types of moist heat. The first is hot water or pasteurization. And pasteurization is usually when we apply beverages, we heat it up to about 60 to 70 degrees Celsius for a period of time. And this helps to kill microbes that are in the beverages that we're drinking, and it helps to increase the shelf life of those products. And so if we're killing most microbes at 60 to 70 degrees Celsius, then hot water is used to disinfect, but it doesn't sterilize. Boiling water is 100 degrees Celsius. All right, my cat is joining us. You may hear some meowing or purring. All right, and 100-degree water, even though it sounds really hot and it should kill a lot more microbes than just hot water or pasteurization, it still is only going to disinfect. So some of those endospores can survive 100-degree Celsius water. Steam heat under pressure. This refers to the autoclave. And so the autoclave is what we put our media in the lab. We close the door and it heats up the liquids to 121 degrees Celsius. And it's under pressure, so it goes up to 15 pounds per square inch, and we do this usually for 15 to 20 minutes. So this combination of heat under pressure and the steam heat that's created really drives that hot moisture into bacterial endospores, and because of that, steam heat under pressure can be used to sterilize. So now that we've gone through all of these different heating methods and you've taken notes in your course pack, you'll want to get out your summary chart and let's record what we've just learned. So here's our chart right here. For our physical and chemical methods, we've got heat. Is it bactericidal, bacteriostatic, or decontamination? And it is in fact bactericidal. And I should have made one block for all of these, because no matter which of these types of heat, it's all going to be bactericidal. Does the method disinfect, sterilize, sanitize, or preserve? Dry heat will sterilize. Moist heat is going to depend on the type. So we won't say anything for moist heat. For pasteurization, this disinfects. For boiling water will disinfect. But steam heat under pressure will definitely kill those endospores, so steam heat under pressure will sterilize. As you go through your notes, one thing that helps to remember these is the exceptions rather than the rule. So pay attention to the methods that sterilize. There are going to be fewer of these than the methods that disinfect. So if you remember the exceptions, it's easier to do that than remember everything. Okay, so do you all remember what the mode of action was for heat? Does it denature or damage all proteins? Does it break up cell membranes, damage DNA, or dehydrate cells? If you recall, it denatures all cellular proteins. But to keep it simple, we'll just say denatures, damages proteins. Okay. So now what I want you to do is go through the remaining physical methods and fill out the rest of your chart for physical methods. So you don't just have heat, we'll have cold temperatures, we'll have radiation like UV and gamma radiation, filtration, and desiccation. I'll quickly walk you through some of this, but I'm going to be taking fewer notes. Feel free to pause as much as you like and take notes when you need to. And you'll also find that some of the notes are in the notes section underneath each slide. So there's more information there. So cold temperatures like refrigeration and freezing. Refrigeration is 4 degrees Celsius, freezing is minus 20 degrees Celsius. Cold temperatures are bacteriostatic. So since they don't kill microorganisms, we're not going to have a mode of action for cold temperatures. It's simply that the enzymes just don't work at cold temperatures. So even freezing, even at minus 20 degrees Celsius, this temperature doesn't necessarily kill microorganisms. So again, refrigeration and freezing are going to be bacteriostatic methods. We don't have any particular mode of action for this. And because they're bacteriostatic, we can use these methods to preserve our foods. And we can preserve foods a lot longer in the freezer than we can in the refrigerator. Some microorganisms, like psychrotrophs or even psychrophiles, can grow at the temperatures of a refrigerator at 4 degrees Celsius. So refrigeration doesn't stop the growth of all microbes, just mesophiles, and it'll slow down the growth of psychrotrophs. But if you've ever pulled moldy food out of the refrigerator, you do know that some microbes can eventually grow in the refrigerator. All right, we've got radiation. There are two different types of radiation. One is gamma radiation, which breaks through barriers. Gamma radiation is a type of ionizing radiation, and gamma radiation will sterilize. So it can pass through barriers, which unfortunately means our skin. So we definitely wouldn't ever want to be surrounded by gamma radiation. It can pass through barriers like multiple layers of meat products, and it causes complete breaks in the DNA. For the mode of action, we're just going to say gamma radiation damages DNA. UV radiation also damages DNA, but it's not as harsh as gamma radiation. What it does is it creates these little mini breaks, not really breaks, but it's a type of damage called thymine dimers. You don't have to write this down. But this type of damage is repairable. And because of that, Non-ionizing radiation, which includes UV radiation, is something that can be used to disinfect rather than sterilize. And because it doesn't break through barriers, it also can only be used to disinfect surfaces. It can't penetrate multiple layers, so it wouldn't be very useful if you were trying to irradiate ground beef. If we're irradiating ground beef, we want to get through all of the layers to make sure that we kill microbes that are mixed up within that ground beef. All right, desiccation or dehydrating. Dehydration or desiccation is when we dry out food products. So freeze drying and desiccation methods will dehydrate our bacterial cells. Therefore, these methods are bacteriostatic and they can be used for preservation of our foods. And our mechanical method that we have on the list is filtration. And so filtration involves the mechanical removal of microbes by forcing either air or liquids through some sort of filter. So if you put a filter in your furnace, it might filter out pollen, or it might filter out even smaller particles like mold spores, bacteria, and even viruses. Really fancy filters like HEPA filters are going to be those that are used in air purifiers, and these can help really sanitize the air. So if you have a water filter, this can filter out some microorganisms. So most filtration methods are all mechanical. They're used for decontamination, and because it's going to remove most microbes, it's used for sanitization. There are a few types of filters that have teeny tiny pore sizes that are actually less than 0.2 micrometers. And these can sterilize liquids. But these are not the types of filters you'll be able to buy at the store. So again, for your sheet, most filters can be used for sanitization. Oops. All right, so now let's move on to our chemical methods. Just like with physical methods, I'll go slowly through the first example, show you how to take some notes in your chart, and then we'll just kind of talk over the remainder of them. So for the chemical methods, what you need to know is fairly similar. The one thing that's going to be different is, can it be used as an antiseptic? So think about things that you use as antiseptics, things like iodine, peroxide, alcohols, things like bleach, no. So use a little bit of common sense and think about what you use in your household when it comes to what can be used as an antiseptic or not. And so as you go through the slides, fill out the notes in your course pack, and then also either during the lecture or at the end, fill out the chemical methods in your supplemental summary chart. All right, so let's start with halogens. Halogens are going to be those chemicals that you find in that second to last column of the periodic table. So it'll be things like chlorine, bromine, fluorine, etc. So fluoride, for example, is something that's used in a lot of toothpaste. And so these are halogens. So our chlorine and bromine, these are going to be something that can be used to disinfect at low concentrations. They're often put in hot tubs and swimming pools. Bleach, this is a chlorite. So on your summary chart, it'll say chlorine and chlorites. These products can actually be used to sterilize, or ST. So these are going to be really harsh and they oxidize all cellular proteins. So we're going to call that damaging proteins. All halogens, for example, cause protein damage. So that will be the mode of action. The mode of action is that they damage cellular proteins. And because this is a very harsh mode of action, halogens like chlorine and chloride and chlorites are going to be bactericidal. Iodine is an example of a halogen, and this is going to be the one halogen that we will use as an antiseptic. So you know that when you go to the hospital, sometimes when you're getting your blood drawn, you get a little bit of iodine swiped over where you're getting that blood drawn. And iodine and chlorites can be sporicidal. So iodine is a very effective antiseptic that's used in hospitals. It may not necessarily sterilize since we're using it on our skin. Remember, it's only an antiseptic if it's being used on skin or living tissues. All right, so let's fill out our chart. So we've taken our notes on halogens as our first chemical method. And now if we look at our chart, I probably should have split some of this up right here. So let's draw, actually, no, undo. Okay, so mode of action. These denature damaged cellular proteins. Really harsh, so they are in fact bactericidal no matter what type. If it's bactericidal, does it disinfect, sterilize, sanitize, or preserve? And so here, I know I said disinfect or sterilize, but I also said if it can be used to sterilize, let's just put that down. So we'll remember that things like bleach, for example, can be used as a sterilant. And then if it can be used as an antiseptic, which means can it be used on living tissues? For chlorine and chlorides, I'm going to say no. For iodine, yes. This is going to be the halogen that we will commonly use as an antiseptic. All right. Aldehydes. Aldehydes include formaldehyde and glutaraldehyde right here. Glutaraldehyde is less toxic, so this is the one that's going to be found in hospital grade sterilants, and it does sterilize. So all aldehydes, they damage proteins, they're bactericidal, and they can be used for sterilization. So that's the key information about those. Glutaraldehyde, less toxic, and if you look at these labels right here, it says bactericidal, sporicidal, virucidal, fungicidal, and tuberculocidal. That means it kills the germ that causes tuberculosis, which is an acid-fast bacterium. All right. Formaldehyde, this is really toxic and carcinogenic, so it's not going to be really used in a lot of disinfectants or sterilants because we don't want to touch any of the residue. Formaldehyde does sterilize, and so it can be used on dead bodies. It's used as an embalming agent. And so what this does is it helps preserve dead bodies by killing all of the microorganisms so that the bodies don't decompose. And so because these sterilize, these are both sporicidal. And actually, if we go back to our chart, I'll fill out one more column for you. Aldehydes, like glutaraldehyde and formaldehyde, also denature proteins. They are also bactericidal. They also are used to sterilize. And because they're pretty dang toxic, we definitely don't want to use them as an antiseptic. Remember, they can be used on dead bodies, but not living ones, especially in the case of formaldehyde. All right, peroxide and alcohols are also damaged proteins. They are also bactericidal. Now we're moving on to things, though, that can be used as antiseptics. So hydrogen peroxide at a low concentration, like you see the kinds that you buy in the store have a very low concentration. This type of hydrogen peroxide can be used to disinfect, and yes, we can use it as an antiseptic. High concentration hydrogen peroxide cannot be used as an antiseptic because it's really toxic at high concentrations. So a high concentration would be about 10% H2O2. Whoa. Okay. everyone. Thank you for your patience. And then alcohols like ethyl alcohol or isopropyl alcohol, also called ethanol or isopropanol, can also disinfect. And again, the optimal concentration for using these is about 60 to 90%. If something is less than 60%, it does not do a very good job. And alcohols are also very commonly used as antiseptics. So remember, even if you have hand sanitizer, it's still an antiseptic because it has that bactericidal agent in it that kills bacteria. Another antiseptic is chlorhexidine. I don't have the mode of action written in here, but chlorhexidine is going to be something that can damage proteins, but it also will break up cell membranes. So two modes of action there. Chlorhexidine is something, it can be used as a disinfectant, but it's often used as an antiseptic. So you'll see it in surgical scrub. You can buy it over the counter at the drugstore if you want to, if you have, for example, stitches that you need to keep clean, you can use chlorhexidine over it. And there's also chlorhexidine, I probably don't recommend this, but you can get mouthwash that contains chlorhexidine. So it would be an antiseptic mouthwash. It'll probably only be used if you have some sort of, oral infection, but something like this is going to kill the good microbes as well as the bad microbes in your house. So chlorhexidine, I think your book calls it a biguanide. We're not going to learn what type of chemical it is an antiseptic. It's definitely, I don't have this written on here, but it's definitely bactericidal. It can be used to disinfect, not sterilize, and it's also used as an antiseptic. So yes to an antiseptic. All right. I'm going to go back to this. I didn't mention this, but when we went to our hydrogen peroxide and alcohols, low concentration of hydrogen peroxide and alcohols, what these have in common is that they both disinfect. Oops. But high strength hydrogen peroxide can be used to sterilize. Chlorhexidine disinfects and can be used as an antiseptic. And then phenolics are also used to disinfect. So phenolics. Phenol is this ringed structure right here with an OH or hydroxyl group attached to it. So anything that has a similar structure to that is called a phenolic. Some are natural, some are commercially produced, but they are added to a lot of household and hospital disinfectants. And some can be used as antiseptics. So these agents, they are bactericidal. They are used to disinfect, not sterilize. Some can be used as antiseptic. So the key word here is only some. So instead of yes or no, it would be a some. And the example is triclosan. All right, and then phenolics, kind of like chlorhexidine, will do damage to cell membranes and cellular proteins. So this would be the mode of action. All right, do I recommend you use these products? The answer is no. An example is triclosan. So triclosan, as you can see here, is called a chlorinated phenolic because of these chlorine groups that are attached to it. There are bacteria that are resistant to these types of compounds. Any type of complex organic compound is going to have a lot of resistance to it. So if it's something that's complex and it mimics something found in nature, there's going to be a lot of resistance to it. All right, so bacteria can become resistant to triclosan. And if triclosan gets in the water, it can actually cause hormone disruption in a lot of aquatic invertebrate animals, little insects. And it can be very toxic, not just to aquatic insects, but also to aquatic bacteria. And also, there's very little evidence that these types of antimicrobial soaps or antibacterial soaps are more effective than regular soap. So if you see a product that has triclosan in it, don't use it. It'll affect your normal microbes, and it's not more effective than regular soap and water. So even right now, as we're worried about coronavirus and using a lot of soaps and antibacterial is not necessarily better. Regular soap and water is very effective. So I don't recommend using products that have these types of phenolics, triclosan being one. So if you are using an antibacterial product, if it's hand soap, then with triclosan, then we would call that an antiseptic. If it's hand soap without triclosan, then we would call it a sanitizing agent or a sanitizer. Same thing with dish soap. If it's dish soap with triclosan or an antibacterial product, then we would call it a disinfectant. If it's dish soap without, then we would call it a sanitizer. But back to this slide right here, these phenolics like triclosan, they can only be used to disinfect and they cannot be used to sterilize. All right, next we have quaternary ammonium compounds. These are also commonly called quats or QACs for quaternary ammonium cations is also what your book might call it. And so what a quaternary ammonium compound is, it has a nitrogen in the middle, just like ammonia is NH4. It has a positive charge, hence the name cation. A cation is a positively charged ion. And then it has 4 groups attached to the side. And they usually have really, really long complex names, like benzalkonium chloride. Even though it says chloride, if you look at the structure, it's got a nitrogen and 4 groups attached. The chloride is just the chlorine salt to make it a negative and a positive charge combined will make it an ionic compound. So even though the word chloride is in the name, it's not a halogen, it's a quaternary ammonium compound. So if you see something like this, it's a quat. Quaternary ammonium compounds are really common in Lysol wipes. So if you look at the ingredients in the Lysol wipes, oftentimes they are these quaternary ammonium compounds. And because they are ionic and hydrophobic over on this end, so you see we've got a hydrophobic region, and then because of the ion, it's also hydrophilic. And things that are both hydrophilic and hydrophobic, we call amphipathic. And because they're amphipathic, they'll lodge into cell membranes. Therefore, the mode of action of these is they break up or they damage cell membranes. So quaternary ammonium compounds, the mode of action is they break up cell membranes. Therefore, they are bactericidal, but they don't kill endospores, so they are used as disinfectants rather than sterilants. They're commonly used as disinfectants. I see very few sanitizers that have, or antiseptics that have these in there, but it is possible that some can be used in antiseptic products, but it's pretty rare. All right. Now we have acids and bases. An example of an acid would be something like vinegar. Vinegar is acetic acid, so it's a weak acid, and ammonia is a base. So acids and bases will also damage proteins, so that is the mode of action right there. They damage proteins, they're bactericidal, but they are not strong enough to sterilize. They can disinfect. Something like hydrochloric acid or nitric acid might be able to sterilize, but let's be real, we're not going to use hydrochloric acid or nitric acid in any of our household products. So for our household acids and bases like vinegar and ammonia, these are going to be used to disinfect, not sterilize. And we're not going to use them as antiseptics. Right. Finally, we've got a couple more to get through. We've got heavy metals. Heavy metals include things like nickel, copper, silver, arsenic, chromium, and heavy metals can be pretty toxic. Because they're pretty toxic, we're rarely going to use them on humans, especially because they can be at high concentrations. They'll not just be bactericidal, but they'll harm our human cells as well. So heavy metals, if they're used in low concentrations, they inhibit enzymes and they're bactericidal. So at low concentrations, heavy metals can be bacteriostatic. They might inhibit enzymes. At high concentrations, however, they're going to be bactericidal because they'll do much more damage to cellular proteins. All right, so bacteriostatic or bactericidal, they are rarely used as disinfectants. Oftentimes, the ones that are bacteriostatic can be used to preserve products. Wood and paint will often have some heavy metals in them to make them bacteriostatic or fungostatic. So a lot of paint for your bathroom will have a fungostatic metal in it so that you won't get mold in the bathroom. And there is going to be very few heavy metals that can be used as an antiseptic. So one example would be these silver nitrate test strips, and you can apply this to an infected wound if the infection isn't responding to antibiotics. So can be applied topically to wounds. Topically means to the surface of your tissues. So whether or not they can be used as an antiseptic, I would be very careful and I would say some, but it's very rare. So an example would be these silver nitrate applicators. Okay, now we have salts and sugars. A high concentration of salt would be salinity. With sugars, we just call that osmolarity. But both salts and sugars can suck the water out of cells, and therefore they are bacteriostatic through dehydration. So salts and sugars, they're bacteriostatic. We use them to preserve foods, and their mode of action is that they dehydrate cells. If cells lose their water, they cannot grow, but they are not dead. Now, last one, we've got soaps. And again, these are going to be soaps that don't contain antibacterial products. So just plain soap. So plain soap products are used for sanitization. They break up dirt and oil. When you scrub soap on your hand, they can also inactivate certain types of viruses like coronavirus, and they physically remove them from your hand. So when you're washing your hands for 20 to 30 seconds, you're scrubbing and you're mechanically removing all of the microbes from the surface of your hands, and then you're washing them down the drain. So these regular soaps, they lack antibacterial products, therefore we don't call them antiseptics. Instead, this is just a method used for hand sanitization. Whether you're sanitizing hands or sanitizing dishes, it's still called sanitization. All right, that concludes all of the physical, mechanical, and chemical methods of antimicrobial control. This table right here just shows you some common products. what they have in them, and then what type of category we would call it. So for example, dimethyl ammonium chloride is one of those quaternary ammonium compounds. If something has sodium hypochlorite in it, that's bleach, and that would be considered a halogen. So this is helpful if you're looking at some of the products you have around the house. Take a look at the label, see what's in it. What type of product is that? What's its mode of action? Can it be used to disinfect or sterilize? So take a look at some of those products you have lying around the household and see if you can apply what you've learned to what you have in your house right now. All right, and so right now everyone's feeling a little bit germophobic because of the coronavirus, but in normal times, these are the things we consider when choosing our methods. Do we want to sterilize or disinfect? Or do we just want to sanitize maybe? So a lot of times in the household, we might just want to sanitize. So vacuuming our floors rather than disinfecting them with bleach. Reusable or disposable. This is going to be more important in hospital settings. So if you are using reusable syringes, you'll autoclave them. But if they're disposable, then you won't need any method to sterilize at all. In a hospital, if something can be treated with high temperatures, then it can go in the autoclave. But if it can't, if it'll melt in the autoclave, then you might have to use UV exposure or some sort of chemical sterilant. So the nature of the object is something that you have to take into consideration. Maybe bleach isn't something that can be used on every object and you'll have to use peroxide instead. If you are sterilizing, you have to know that the agent will penetrate biofilms. All right, this isn't really applicable to coronavirus because coronavirus doesn't grow in biofilms. But if you are sterilizing an old catheter in a hospital lab, then you want to make sure that the entire biofilms that might have grown on that catheter are penetrated. And at home, you want to make sure the agent is cost effective, and you also want to make sure that you don't use products that contribute to antimicrobial resistance. So again, if you use products like phenolics or quaternary ammonium compounds, right? If bacteria are resistant to these antimicrobial compounds, then they may be resistant to other antibiotics as well. And so sometimes when we use these products, what we're really doing is we're selecting for bacteria that are resistant, not just to those products, but possibly others as well. That are resistant. And so what products should we use? If you don't want to use these, what you can use would be something like bleach if you need to disinfect. You can use ammonia. You can use alcohol. You can use hydrogen peroxide. Or don't forget, you can also just use soap and water. So some of my tips that I have for the household right now. Don't use those products with complex chemicals that bacteria have developed resistance to. Unfortunately, this includes wipes. Right now, wipes are being bought a lot because they're very handy. And right now, people are really worried about surfaces and coronavirus. So if you're using disinfecting wipes right now, don't worry about it. I won't judge you. I'm using some of them too. But on a regular basis, when we're not worried about a pandemic, try to just clean with soap and water or something like alcohol or ammonia. And this will work really well, and it doesn't contain any of those complex chemicals. If you're just trying to sanitize, right, like most surfaces can be sanitized. You can use filtration, like a vacuum filter. You can use soap and water if you're cleaning surfaces. If you need to disinfect, so think about a kitchen counter after you've cooked with raw meat. You want to make sure you use something to disinfect so that you can kill any salmonella that might be left behind. Alcohol, vinegar, or ammonia. If you just buy some of these compounds from the store, put them in a spray bottle, dilute them to their optimal concentration, it's really cheap and it won't promote resistance amongst the bacteria on that surface. Um, and then finally, if you need to sterilize in your household, think about your bathroom. Think about the places that poop gets. These are the locations you want to sterilize. And so for these types of products, our bathroom cleaners or our toilet cleaners, we're going to really want to make sure we use something with bleach or hydrogen peroxide. In the household, we're probably not going to find products that contain glutaraldehyde. Those are typically only used in hospitals, but these are going to be our more common household products that can be used to sterilize. And so I hope you've learned something today. I'm going to go ahead and bring up our course pack one more time here. All right, so at the end of this lecture, you should be able to go through all these physical and chemical methods of microbial control. You should know their modes of action, whether they're physical, chemical, or mechanical, whether they're bactericidal or static, whether they're used to disinfect, sterilize, sanitize, or preserve, and if it can be used as an antiseptic. And towards the end of this week, what I'm going to be doing is I'm probably going to give you a quiz or activity related to this bullet point right here. Look for it on Blackboard on Friday. I'll also send out an announcement when this is available. And so you have, hopefully already by this point, done the adaptive Learn Smart reading assignment. Next week, I'll give you a follow-up McGraw-Hill homework assignment where I've chosen the questions and you can answer them. And on Friday, I'll also post a short quiz where I give you some different scenarios and I'm going to ask you for some physical or chemical methods you might be able to use. So that type of quiz will involve applying your knowledge rather than just memorizing some of the information. If you have any questions, please feel free to email me, or we can start a new discussion board thread, or we can create a Zoom live lecture to answer your questions. All right, with that, I'm signing off. Thanks all.